central question in biology. What is the minimal unit of replication? Right? Biology is all about self-replication. Cell makes a copy of itself, DNA makes a copy of itself. So, what is the minimal unit of biological replication? Any ideas? Two what? Oh, you're saying a factor of 2. No, I am saying what is the minimum machinery you need to do self-replication? What is the minimum molecular machinery you need to do self-replication? Okay, DNA. Can DNA self-replicate? DNA can't self-replicate, good. So, this idea that DNA is the key to replication is not fully thought through, right, because it needs more. Okay. So, if it is not just DNA, then sorry, cell is the minimal one. You can't imagine anything less than a cell that can self-replicate. Little louder. Some organelles can self-replicate. Self-replicate. I mean, I am asking a very strong condition, right? Can I take an organelle out, put it in a tube, and have it self-replicate? You are saying some organelles can replicate, and you are correct, but can they self-replicate? Definitely, a cell can self-replicate. I put it in a bunch of stuff, and it will replicate, right? But think again. Okay, in fact, while we are at it, let me pull out another slide, which is not in this talk, but which will be useful to think about. But just keep talking. So, what suppose you as an experimental biologist had to make a collection of molecules in a tube that was fully capable of self-replication. Would you be able to do it? If so, how? Okay, you need DNA polymerase. Anything else? But okay, fine. Keep going. So, that's enough to self-replicate? You might be right. So what will that do? So, if you put nucleotides, yeah. So, you are talking about PCR, right? Yeah. So, the PCR reaction, of course, there is a problem because it does not make the polymerase. You have to add the polymerase protein, right. So, when I say self-replicate, I am saying you take sort of ordinary stuff that you might find on any meteor or any comet, right, and put it in a tube and then somehow have some ability to self-replicate. So, I am just going to load up some new slides here. Does it work? Yeah. Here we go. So, try again. Okay, you are right. That starts to get you on your way. If you put DNA and you put nucleotides and you put some energy, you will start to get something, but you have to put that protein, the Taq polymerase. So, to make that protein, what do you have to do? You need the gene for that protein. Okay. So, if you need the gene for that protein, but that gene on its own can't self-replicate. So, the gene has to be transcribed. So, you need a whole ribosome. So, how do you make a ribosome? You need like 100, you need to have a whole translation machinery. Okay. So, try again. What is the minimal unit? Okay. It is an open question whether the minimal unit of self-replication is an entire cell. It is certainly a cell is a unit of self-replication, right, but whether it is an entire cell or something smaller. Okay. So, here is a thought experiment. Okay. So, in this flask, I put sugar, salt and water. I literally mean that. Of course, when I say salt, I mean salts, sodium chloride, ammonium sulfate, and a bunch of different salts. So, I should have said sugar, salts and water, but literally I will put in only that. When I say sugar, I mean sucrose. Okay. When I say salt, I mean stuff you can buy or dig up out of the ground. Okay. And I have a pump, and the pump flows all that stuff into this black box. Then I have another pump which just takes the same stuff out of this black box and puts it into another flask. So, clearly all this system is doing is moving sugar, salt and water from this side to this side. Okay. Now, I do something magical, right? And by doing something magical, I have a system where I take sugar and salt and water and put it into this black box, and then what comes out of this black box are living cells, right? Does anybody think this is possible? How many people think this is possible? How many people think this is not possible? You're either on one side or the other, you can't sit it out. How many people think this is not possible? Some people. Okay, everybody else thinks it is possible. Okay, so for all those of you who think it is possible, what did I do here? That's how life evolved. Sure, there was one, okay, fine, so very good. So of course, at some point in time there was not any living things, and at some point in time there are living things, right? So at least we assume that there was a time on Earth, before the Earth formed there was no life on Earth anyway, and after a while life evolved. And the current sort of scientific consensus is that life evolved on Earth, although there are some people who think it came from somewhere else and landed on Earth, right? But then it must have evolved somewhere else, right? So whatever it is, there must have been a planet on which life evolved, right? So, okay, that's at that time scale. I am asking, in the lab, can I do this? I am not asking, can I put sugar, salt and water and wait 2 billion years? Right? By the way, how old is the Earth? About 5 billion years. And how old is life? About 3 or 3.5 billion years, pushed back by fossil evidence. Okay? So, in some sense, very soon after the Earth formed, we have had living things. Certainly, the origin of life is closer to the origin of the Earth than it is to the present day. Okay, so anybody knows what I did with this box? Come on. Everybody here who didn't raise their hand saying it was impossible, obviously you believe it's possible. So now you can tell me what I did there. So let's try again. I want you to raise your hand for one side or the other of this question, not like this, hoping that nobody sees you, but you have to raise your hand. How many people think this is impossible? It can't be done. Raise your hand. Don't do it like this, right? So I can see it from here. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. How many people believe this is possible? Can be done? 2, 3, 4. Okay. How do you think it can happen? But if you had to do it in the lab, right? Okay. So whatever you might think or not think, this is pretty astonishing, right? I have a system that is just a pump that takes sugar, salt and water and moves it from this side to this side. Now it's exactly the same system except in this black box. I have a pump that takes sugar, salt and water, brings it in here, pulls it out, and now I have a machine. So here I had a machine that just moved things around. Now I have a machine that takes sugar, salt and water and makes living cells. Okay, so you're thinking I'll put amino acids, electricity and high pressure and temperature and get living cells out, right? It's possible. Yeah? There you go. Okay. It's obvious, right? Now this is the thing, right? If I just shown you this, then all of you will say, yeah, that's totally obvious. So you are willing, when biology does something totally ridiculous, you'll be like, yeah, that's fine, because it's biology, it doesn't have to obey any rules that you're previously used to. That's the attitude, that if biology does something that looks like magic, you're willing to let it slide. Like, that's okay, because it's biology, it doesn't have to satisfy all the other things I believe the universe has to satisfy. If it's biology, I don't hold it to that kind of thing, and you shouldn't do that, right? Because biology is made of molecules. Molecules obey physical laws, right? So there's no reason that biology should escape from being constrained by the physical laws, right? So this experiment, just like I said yesterday, unless you have an expectation of an experiment, you're not going to be surprised or astonished by it, right? So this, I find, as a thought experiment, is pretty astonishing, right? I took a system that was just a pump that moved sugar, salt and water from one side of the room to the other, and I've converted it to a system that moves sugar, salt and water from one side of the room and ends up with living cells on the other. So I have, but now that you see how it works, it's easy. Yeah.
Where do you get the nitrates and phosphates and other things?
So the salts contain, the salts will be something nitrate, something phosphate. It's not just NaC. That's what I said. When I said salts, I mean a bunch of salts. Yeah? So I should have said salts. But it's, simple inorganic salts. Right? The only carbon source is glucose or sucrose. Okay. So now if you imagine this, now imagine what that 1 micron sized cell has managed to do. The 1 micron cubed sized cell has managed to initiate a collection of reactions in this flask that takes at the atomic level all the stuff that comes in here and starts putting them together piece by piece, block by block, into a structure which is another copy of itself, and then more and more and more copies of itself, and so on. Right? It's incredible. That tiny thing. Right? So the other way I state this problem, because people are not astonished enough by this, is I ask them, when you buy a bag of atta from Food World, it's totally vegetarian, it's sealed from the environment, no air goes in or out, right? But if you wait 3 months, it becomes a bag of flour beetles. But now, how does a bag of flour become a bag of flour beetles? Where do the flour beetles come from? Everybody's seen this, right? If you don't open your bag of atta, how many people have never walked into their home kitchen here? Maybe some people never go into their kitchen, okay. But if you've ever been to a kitchen, right, and you've ever had to shop for your own atta, you know that if you don't use it, it'll be converted into some other kind of stuff, right. So how does a bag of atta become a bag of flour beetles? Where was the information to make flour beetles? The atta can't spontaneously become a flour beetle, right. So how does it know? Where is the information? Raise your hand, I'll come to you. I can't hear or see. It's very loud in here, so if somebody says something, I can't quite see who's saying it. Somebody there said something? Moisture. Spontaneously. You mean flour plus moisture can make flour beetles? Why didn't it become dinosaur? The chemical structure of flour forces it to become a flour beetle. Yeah. Yeah. When you say earlier form, what do you mean? Egg. Okay, fine. Not so surprising, right? Can is one egg enough? Yeah. Sure. Yeah, sure. Obviously, there were some eggs in there. It's not that, guys, come on, flour can't spontaneously become a flour beetle. Obviously, there was an egg in there, right? Yeah. One egg is not enough, as it happens, because flour beetles are sexually reproducing. So you need at least 2 eggs. And if one happens to be male, one happens to be female, and they start to mate and so on. So always there are a few eggs, right? Now my question is, was that bag of flour at the beginning vegetarian or non-vegetarian? Right? In this day and age, this is the big question, right? How many people think the bag of flour was vegetarian in the beginning? If a bag of atta is not vegetarian, then what kind of world are we living in? So surely the bag of atta, how many people believe a bag of atta you buy at Food World or whatever is vegetarian? Nobody believes that. Sorry? No. I'm talking about you really go and buy for your grandmother a bag of atta. You're going to tell her this is a non-vegetarian atta, is it? No, come on. I'm asking a real world question. Bag of atta is vegetarian, obviously. Are there some of you who believe a bag of plain flour is not vegetarian? This is a yes/no question, guys. Okay, you're saying it's not. Is there anything that's vegetarian then? Is there anything that's vegetarian? They coexist, okay. So it's 99% vegetarian, 1% non-vegetarian. Okay, fine. So he's, okay. But in the real world, I'm not asking you guys to answer a legal question. In the real world, if you go to a restaurant and ask for something vegetarian, have they bought some special atta where they've removed some eggs? Is that what they did? Okay, so obviously the atta is vegetarian. How many people believe that eating beetles is vegetarian? How many of you have eaten a beetle? Any of you have eaten a beetle? No, never. You've never bitten into a fruit and seen half an insect? Okay. Anyway, so when did that bag become non-vegetarian from vegetarian? That's why he's worried now. I put him in a position where I have to posit that non-vegetarian stuff can arise out of nothing, right? That's why he got worried. But obviously it can, right? Obviously, a bag of vegetarian atta can become non-vegetarian at some point, right? And the reason is because there's at least 1 or 2 eggs in there, and what's in the eggs? It's the genome of those beetles, right? And what's in the genome? It codes for the species. And when that beetle larva is formed, what does it do? That little beetle larva, what does it do? What's the first thing it does? It eats the atta, right? And then it breaks the molecules of the atta down into building blocks, and the same building blocks that make atta also make the beetle, right? Isn't that true? It's not like it contains new elements, yeah, and all it does is rearrange. So, the beetle DNA through a chain of events causes the molecules in the atta to be broken down and to be rearranged to form a beetle. It's obvious, right? So, where is the line between vegetarian and non-vegetarian? At what point in that process would you say, right, when half the bag is beetles and half the bag is flower, or when? Or as you said, right at the beginning. If it's right at the beginning, you've put yourself in an absurd position. You can't live in this kind of system where you're willing to make ridiculous answers to scientific questions, but not use the same kind of logic in the real world, right? That's the main problem with scientists. They think what they're working on has got nothing to do with real life. Right? If you're living in that kind of situation, you shouldn't be doing science. Right? Science is about real life. So, I use this example because people are very happy to assume that non-living things can become living things. They are not happy to assume vegetarian things can become non-vegetarian things.
Eating the plant cell is
Yes,
vegetarian,
sure.
the animal cell is not.
Very good. And at some point, we've just taken plant cells and made them into animal cells, right? So, one can become the other. Yeah. By the way, most vegetarians, particularly in India, get all their trace organic nutrients because of contamination by insects in flower and fruit. If we removed all the insects from the food, many people would get nutrition deficiency diseases. Okay? I mean, don't tell your grandmothers, but this is true. So, it's all there. Okay. So, this is just a thought experiment to set up the big question, right, which is this astonishing thing that a 1 micron cubed object can set up a chain of reactions which allow molecules to be broken into tiny bits and rearranged to form a living cell. Same molecules are non-living, same molecules can be living, right. And now I am asking you, what is the minimal collection of things that you need to make that happen? I can add 1 living cell and then it becomes totally obvious. Can I add anything less than a living cell and make this work? Right? And let us say I don't want living cells out here, I just want new copies of what's in here coming out. Like people often say crystallization, right? Crystals make copies of themselves, right? Is that the same thing? You might want to think about it. Okay. So, no, but so in my group, we generally think about these 3 categories of questions. So, everything I work on is at the level of cells, right? I don't work on organisms living in the environment much bigger than a unicellular organism. I do think of cells in big organisms, but not think of the big organisms themselves. But think about how cells evolved, and we won't get to that question today. But certainly, I think where is the information encoded for a cell to make another copy of itself? Right? Where is the information? So, as I said, that little in this experiment, what species of cell will come out here? The same one as went in here, right? Now, suppose I took the cell in here, I took the same cell and I managed to use a little micro injection to pull out its DNA, and I put in the DNA of some other species into the same cell. What species would come out here? Suppose I took cell A and then I pulled the DNA out of cell A and I put the DNA from cell B into the same cell. What would come here? B or A? How many people think species B would come out? B is the one that I put in by the DNA. Okay, how many think cell A would come out? Nobody. I take cell A, let us say I take E. coli, okay, and then somehow using little manipulation I pull out the chromosome from that cell and I put in the chromosome of Salmonella into what used to be an E. coli cell, and that is what is in here. What will come out? E. coli, the original cell, or Salmonella? Okay, yeah, that is right. So, somehow the information at least is in the DNA. The DNA can't self-replicate, right. So, this experiment has been done. It has not been done with E. coli and Salmonella, but it has been done with a mycobacterial species. So, Craig Venter did exactly this. Right. He took a bacterial cell, pulled out the DNA of that species, put it in another species, and what happened was the cell became the species according to the DNA that it contained. Right. Now, of course, you can't take a bacterial cell, put in the human genome and get a human, and the reason is because you can think of the bacterial cell as like a, I do not know if even now, I do not know how many of you actually have seen a DVD player, right. Okay, it is not as old as I thought, right. So, the DVD player is like the cell and the DVD is like the genome, right. The DVD on its own can't play a movie, right. The DVD player can play many movies. The movie that comes out on your TV screen is the movie that you put in on the DVD. Right. So, the DVD itself is not fully capable, but the DVD player plus the DVD is the full system. Now, the problem with that of course, is that the DVD player can't make copies of itself. DVD player can make movies, right, but a cell can make copies of itself. So, it really is the genome that is making these things happen, right. So, the information of which cell you are making is contained in the DNA, but that does not mean DNA is fully capable of self-replicating. You need the information reader, right. You need the information reader, and the evolutionary gap between humans and bacteria is so large that the human reader system and the bacterial reader system are very different, right. So, that is why you can't put a human chromosome into a bacterium in the same way that you can't put a DVD into a cassette player. How many of you have seen a cassette player? Okay. So, you can't put a DVD into a cassette player, right, even though they are both sort of conceptually the same kind of thing, right. So, that is why, but in principle this kind of experiment could be done. You could, for example, put a mammoth genome into an elephant egg, and what you expect to come out is a mammoth, right, and there are people talking about doing these kinds of experiments. But these experiments are actually being planned as we speak. Okay. So, when I say information to make the cell, right, do I mean information in the same sense as computer science, or is biological information somehow different? Well, it's not very different, right. So, as Kavita said on the first day, when Darwin had this insight about where all the variation of life on earth comes from, he was not basing it on a particular model of where the information is contained. Why does heredity work? Why do children look like their parents? He had ideas, but it was not central to his theory, right. But by the time Mendel came along, you knew, they were actually around at the same time, but Mendel's experiments showed that there was, at least in some situations, what seemed to behave like tiny particles that were inherited from parents to offspring, and those tiny particles somehow contained the capacity, astonishingly, to change an entire macroscopic organism. Okay. How small were those particles? Well, you could actually compute, right. You could say how big is an egg, right, and how many different traits does it encode, and you can break up the volume of the egg into all those traits. You could work out a particle could not be very large. So, whatever that tiny particle was, it contained information to have an impact on a large scale, right. That is one of the things biology does. It converts small things into big things. Yeah. Now, in parallel to the discoveries of the molecular basis of this in the 20th century, the DNA, translation, genetic code and all that stuff, right. In parallel, the computer scientists, so this is Shannon, who invented information theory, and this is Turing, who invented the idea of the computer. They were thinking about informational devices, but they were thinking about it for other purposes, for making phone calls, for breaking codes, right, during World War II, for example. But it turned out that they both had roughly the same kind of idea. So, just like information in your DVD is zeros and ones, right, obviously we have heard it many times, that information in your DNA is also digital in the sense that it is discrete. Instead of 0 1 is ATGC, but that is not really a fundamental thing, which is why when you do models of population genetics, you can without loss of generality assume it is just zeros and ones, right, because conceptually exactly the same thing. Now, how do those zeros and ones convert to an animal? We do not know the full story. We know how it makes proteins, but that is just the starting point, right? How the proteins get together to make the egg develop and make the embryo, the fetus, and this is all stuff that is very big, right? Okay. So, in terms of how much information there is, right? Everybody knows the unit of information, right? Before Reliance Jio, everybody knew the unit of information because you had to pay for it. Because now nobody has to pay for it, so nobody keeps track of it, right? But a bit is a unit of information. What is a bit? It's a 0 or a 1, right? But the units we use in the practical world is something called a byte, which is 8 bits. Everybody knows a byte is 8 bits, right? Something you learn. Why a byte? Why not just a bit? So it turns out a byte is just enough to encode a letter or a character, right? There's something called the ASCII code. So all the lowercase and the uppercase English letters, all the numbers, all the commas, all the funny characters, there's 256 of them, so you need 8 bits to make a letter. So the unit of a nice, easy unit of information is 1 letter, right? So if you think of a bit as about 1/8 or 1, I'm just using units of 10, right? 1/10 of a byte, right, is a yes, no answer. 1 byte is a letter. 1 word is about 10 letters, give or take, right? On a printed page, you can have about 1,000 letters, right? So 1 kilobyte. A gene has just a few, right? You don't need that many English letters to encode a gene. A photograph is much bigger than a printed page, right? A picture is worth, in this case, 100 printed pages, much more than 1,000 words. All the works of Shakespeare, 5 MB, and the entire bacterial genome can be compressed to less than 1,000 or 1 million letters. Right? A single song is much bigger, a CD is much bigger, but a human is just a couple of CDs. Just a couple of CDs, not even a movie. Right? So the line I give is, it takes more information to encode a Rajinikanth movie than to encode Rajinikanth himself. But how can that be? That's crazy, right? But this is true. So we're still trying to figure it out, right? So where is the information? Yeah. So again, I asked, all life is made of cells, but how is a cell made and how does one cell make another cell? Now somebody already brought this up. There's a very famous experiment, Miller and Urey experiment, already very old now, classic. They were looking at spontaneous generation of life, right? And of course, they never saw that. Nobody's ever seen spontaneous generation of life. But they certainly were able to spontaneously generate complex organic molecules from simpler organic molecules, right? They put these things in a tube, they zap them with electrical sparks, there was high pressure, all kinds of toxic gases, and somehow you made complex organic molecules. Now it turns out, we know today, just by looking at the light that comes to us from the sort of rocks that fall into Earth from space, that a large proportion of the solar system in the form of these orbiting asteroids and more distant objects contain these organic molecules. The organic molecules are around, right? Even things like nucleotides, amino acids and so on, you can find them in space. Now that doesn't mean you get life out of it. Now one of the first people to really grasp the difficulty of this problem, right? So Mendel had thought about the gene, and that's all totally fine, right? But the idea that the gene itself is not a self-replicating entity, right? So just understanding what a gene is doesn't help you understand what life is. This is a big mistake molecular biologists make. They think everything is genes and proteins, and that's it, and that's enough to explain everything. I mean, it's certainly, obviously, everything must be explainable from there, but it doesn't mean we have an explanation. And it doesn't mean we can throw up our hands and say, beyond this, we can't explain it. We should try to explain it. Right? So von Neumann, how many of you have heard of von Neumann? So he's one of the most prolific, influential scientists of the 20th century. Right? So he worked in quantum mechanics, information theory, all kinds of applied work. And apart from everything else, he wrote this book, which is called The Theory of Self-Reproducing Automata. So he was really imagining, what does it take for an object to just take the building blocks in the environment and make a copy of itself, right? So instead of a DVD player, you can imagine, can you build a 3D printer that can make another 3D printer? Is it possible? And he was thinking about it purely concept. What does it need? It will need an information storage device, right? It will need a device that makes a physical apparatus. It will need, error checking. I mean, he just thought about, just like you think about the pieces of a computer, the CPU, the memory, and so on, right? He was thinking about the pieces you need to make a self-reproducing entity, not necessarily a living thing. Now, these kinds of ideas were taken by another very famous physicist, Freeman Dyson, and he asked, can I imagine doing this at the molecular scale? Not a big 3D printer, but can I imagine making a very, very tiny device that's based on simple laws of physics and chemistry? What does it take? Yeah. So, as it happens, a very influential paper of this type, my colleague Sandeep, when he was doing his PhD with Sanjay at IIC at the time, the idea they had was that there were lots of chemicals, and these chemicals could convert other chemicals into third types of chemicals, right. So, there was no distinction between what is an enzyme, what is a molecule, or whatever. Everything is making everything else. And if you have a random pool of prebiotic life and molecules were just driving chemical reactions that converted building blocks to bigger things, and those bigger things were used to convert something else into an even bigger thing, and so on. They made these random networks, and they found that in those networks there were these little very self-referential components which they called autocatalytic sets. Autocatalytic means they are a positive feedback reaction that as soon as you have enough of these, they start to make more and more of these by using up all the rest. So, this is sort of a picture of a prebiotic environment that can use building blocks and make more. So, this indeed is an example of a self-replicating system. You could argue that it is a very simple one, but actually it contains a lot of the ingredients that might correspond to what a living cell does. Okay. And in principle, you could imagine making a setup in a test tube that does something like this. Okay. Now, to do it, of course, you need to make sure all the chemical reactions happen in the right direction and so on, but these days we have the power to make small molecules that have designed properties. So, there are people doing this kind of stuff. Right. It's an open field of sort of chemical and biological exploration. So, this is all very abstract, and there were lots of people who were wondering literally how does the cell do it? What is the minimal unit of cellular replication? Right. So, Lvov, Monod, Jacob, these were pioneers of 20th century microbiology. This is a very beautiful picture because these dice represent Monod's idea that there is this unit of randomness inside the living cell, as we have been discussing. So, they thought if we want to understand how a cell makes two cells, right, you can't just sit in the armchair and think about it, and it is also not sufficient to break up a cell and see what molecules are in there. Right. If I break up a cell and I get the molecules out, it is not that I can put all of them back in and suddenly it makes a cell. It just does not work like that. Right. It is not just the molecules, it is the processes that are important. So, once you have broken the processes, you can't make them again. You will have to put all the molecules back with the correct velocity and the correct orientation. There is no way you can do that. But the entropy is already gone up. So, they said let us just watch how a cell grows. Okay. How many of you have actually been in a biology lab? Yeah, and have you done a bacterial growth experiment? Any of you? 2 people? 3, 4 people have done a bacterial growth experiment. It is the most trivial experiment in the world, right? Very, very easy. So, when you do a bacterial growth experiment, what happens is you put one cell into a tube, and then you just track how many living cells there are as a function of time, and then when it exceeds the capacity of the tube, you redilute it. Right? You take a little bit, put it into another tube, and then you with new food, right? Then you watch it grow again, right? Because exponential growth, which is what I am coming to here, is so fast that you very, very quickly use up all the food, and then you can stitch all these together to make what would have been the true exponential growth profile, right? So, in general, what you find is that any bacterial species, when you put it into a tube and there is sufficient amount of food, right? You are not running up against the saturation limit that we have heard about in other lectures, then a standard thing happens. 1 cell becomes 2, 2 cells become 4, 4 cells become 8, and so on and so forth. They might get desynchronized a little bit, so they do not happen in bursts. So, when you watch and you look, you do not get geometric growth, you do not get 1, 2, 4, 8, 16, but you get a more continuous version, exponential growth, just because it gets desynchronized. Otherwise, you might have even seen geometric growth, right. So, it is very simple. Why do cells grow exponentially? Just because 1 cell becomes 2, okay, very simple. That is not why exponential growth is interesting. This is sort of totally obvious, right? But it turns out that once a cell is growing exponentially, once a population of cells are growing exponentially, you can take a cell on this day or on that day or on this day, and they will have, at the moment of division, they will have pretty much the same chemical constituents, right. So, exponential growth is a steady state, it is called balanced growth. Now, it turns out that by measuring the growth rate, so how do you plot exponential growth? You do it by plotting time on this axis and plotting log of the number of cells on that axis, and then you get a straight line, right. So, this is the reason why in biology exponential growth is known as logarithmic growth. It's perverse, but that is what it is, right. So, it is actually not logarithmic growth, right, the growth is exponential. It so happens that if you plot log as a function of time, you get straight lines. You can capture the slope of that straight line, and that slope is the exponent. So, you can say that the number of bacterial cells is e to the gamma t. We have seen this also many times over the past week, right. And you can take that value of gamma or lambda in this case, the growth rate, and you can put it under conditions of various types of food sources and various types of antibiotics and so on. And it turns out that everything falls onto very well defined families of lines, and by looking at these lines, you can start to understand a huge amount about how a bacterium uses nutrients, how it breaks up nutrients and so on. So, I will not go into this further, but you can certainly read these authors or more. But where I started getting interested in this is something that has been known since easily the 1970s, although the paper here I have shown is a sort of review from 1990. So, what is being done here is this is time, this is log. So, it is log base 10 of some sort of estimate of the number of living cells in the tube. I will tell you in a second how you measure that. Assume this is correct, right. So, log 10, the CFU means colony forming units, it does not matter what that is, it means number of living cells per ml of the tube. And what you find is that cells will grow in the absence of an antibiotic, but as you add the antibiotic, the growth rate slows down, and as you add more antibiotic, the growth rate actually goes negative. So, how does growth rate go negative? Instead of e to the plus gamma t, it becomes e to the minus gamma t. Instead of exponential growth, you get exponential decay, right. So, this is very reminiscent of in radioactivity. So, in fact, it is exactly equivalent. So, this is a exponential decay. In radioactivity, if you plot the activity of some radioactive sample as the nuclei decay and you pick it up on a Geiger counter or something like that, you can measure the activity, it drops in a very characteristic way. If you plot the logarithm of the activity as a function of time, you get a perfect straight line, right. This is nitrogen 16. Right. So, the real question that came to me was when looking at this stuff, which was not in the textbooks, right. So, exponential growth is very easy, 1 becomes 2, becomes 4, becomes 8, right, that is totally obvious, because that is what cells do. But what does exponential decay mean? Right. So, can somebody explain why you expect a curve that goes down like this? What kind of curve would you have expected? If you add suppose instead of bacteria, right, I had I do not know, worms, right, and I took a tube of worms, I know the number of living worms, and at some point I add some poison, and I plot the number of worms as a function of time that are still alive. What would you expect that curve to look like? So, in the first 2 minutes, they will all still be alive, right, and then what happens? What? Sudden, yeah, at about 10 minutes, maybe they will all die, but by the time it hits their nervous system and, yeah. So, this will happen with any complex organism, right. So, I mean it certainly will happen with humans, unfortunately. So, when somebody is suffering from a disease, what happens is you survive for a while and then you sort of succumb to it. Now, even among whether it is humans or mice or worms or whatever it is, there is still some variation, right. Not all the worms will die at exactly the 10-minute point, right. Some might die at the 8-minute point, some might eke out some survival till 10, maybe even 15 minutes. But why is that? That is because there was some variation in the health of the worms to begin with. Right. Some happened to be more healthy, they had more energy reserves, and so they were able to resist the poison slightly longer. So, they will die later, these will die earlier. Right. But when you plot that curve, it will be some complicated curve, right. Some were healthy, some were not healthy, you do not know. Why would the curve of the number of worms as a function of time be a perfect exponential decay, which is what it is here? There is no reason. Yes. Sure. Now, we have very good. We have to start thinking about what you see. What this implies is that some cells have died here, but there is still some more alive, right. some different cells died here, some different cells died here, some different cells died here. So, what are the possible ways we can think about this? Either there was variation between the cells to begin with, but if there was variation, that does not really help us understand why it is such a simple mathematical law, or some other crazy thing that the cells are all almost the same, but they start competing for food, and whichever one survives, it manages to live for another couple of hours, competes again, whichever one survives, right. It is like a knockout round in a tennis match or something, right. It is possible, it is possible. In fact, that is a reasonable hypothesis which one can test. So, now it comes down to, can you build, so this is the context in which can you take a theory that you know about, can you build a model at least to try and understand this, because on the face of it, it seems very mysterious. And then our job will be to build many models which contain many hypotheses, right, including competition for food, including initial variation, and just test those models against the reality, right. So, the theory, or at least the big picture that comes out from this is the following thing. So, when the number of living cells is growing up, is going upwards, right. So, this a line with positive slope is growth. Macroscopically, I can watch it because I can see the number of cells in total going up using a macroscopic measurement, right. And if this slope is negative, we call it decay, just like radioactive decay. So, growth and decay. So, growth and decay are not directly related to the microscopic processes that the cells are actually undergoing, right. Microscopically, what is happening to a single cell does not care about the whole population. A single cell has just one choice to make, either it is going to die or it is going to divide, right. It can die or it can divide. And so, roughly the model, any model we have to have in mind, right, must take into account that these are the microscopic processes, and we have to see how these relate to the macroscopic observation. So, roughly the idea is that if you see growth, then you will guess that division is greater than death. The division rate, the division probability per unit time is greater than the death rate, right. But if you see decay, roughly the division rate is low and the death rate is high. Of course, the death rate may be 0 here. I am just putting it up there as a possibility, division rate may be 0. And I can't tell the difference between all these, because when I see this, I do not know how many dead cells there are, right. I need to separately measure division and death rates, which means I must be able to separately measure the number of living and the number of dead cells. It turns out I can do that, it is very simple. So, the way I measure living and dead cells separately, first of all I measure the living cells. How do you measure the living cells? You take a flask, you take out the cells in a pipette, you take a fraction of a cc of it, right, let us say 50 microliters, and you put it onto a petri dish, and it is so dilute that all the cells land in a separate part of the dish, and there is so much food over here that if the cells are alive, they will grow into a little colony. So, when I come back in the morning, the number of colonies I see here is the number of living cells there were in that unit volume, right. That is why they are called colony forming units. And if I know the volume of the original pipette sample, then I know the density of living cells, right. So, this is how I measure living cells. How do I measure total cells? I use a completely different approach. Now, whether a cell is alive or it is died, it will still scatter light, right. So, I shine light through this at 600 nanometers, and cells will absorb at this light and they will scatter, and by measuring how much they absorb, I can work out something called the optical density, right. And then separately I can make a calibration between optical density and number of cells by just taking a sample with just living cells and doing this experiment twice, right. So, this is the calibration unit which we can measure in the lab, right. So, if you measure this blurriness of the flask multiplied by 8 times 10 to the 7, you will get the total number of cells in the flask, living or dead. Okay. And then by subtracting this from this, you will get the total number of dead cells. Huh? Very easy. In practice, it is actually hard to do these experiments carefully enough to make it useful, but you can do it. So, here is the data. Let us remember what I told you, total cells and living cells. So, in all these plots, there are two curves. The sort of lighter version of the curve will be total cells, and the darker version of the curve will be the living cells. Right. Now, what have I done here? I have shown you the result of 3 different experiments. And the only difference between them is that I add an antibiotic. There is an antibiotic called kanamycin, it does not matter what it is, but there is none of it here, there is some amount of it here, and there is much more of it here. So, let us just take this case, but there is none of it. So, here until the food runs out, you have perfect logarithmic exponential, you have perfect exponential growth over several hours, right? And the total number of cells is basically all living cells. Okay? They track each other perfectly. This is how I got that scale factor. Okay? And the picture you should have in mind is 1 cell becomes 2, 2 cells become 4, 4 cells become 8, maybe 1 cell dies. After it dies, it's just present as a carcass, but they are a very tiny minority of the total tube, clearly. What happens when I put a huge amount of antibiotic? When I put a huge amount of antibiotic, initially, just like those worms, the cells manage to survive, right? But eventually, they all die. And once a cell is dead, it does not make 2 copies. This cell had 2 daughters which are both dead, and beyond that nothing happens, right. So, once a cell is dead, it just keeps going as a single line, it does not go as an exponentially doubling line. Right. So, once all cells are dead, the total number of cells becomes constant. So, that is what happens here. Initially, the cells do divide, but then they start to die, and they die exponentially, and eventually the total number of cells will all be dead cells, there will be no living cells. If there are no living cells, you cannot make any more cells, the number of cells total becomes flat, right. So, that is the difference between here, both the lines are the same, here this becomes flat, that goes to exponential decay. The really interesting thing is what happens when you just manage to find a balance, right, where just enough cells for some reason that we do not really know yet manage to survive, right. In this case, every time a cell divides, the chance that one of the daughters is dead is half. But once a daughter has died, they will just continue as a carcass, but there will still be the same number of living cells as they were to begin with, because on average every living cell makes only 1 copy of itself. Right. So, that is exactly what happens. I mean, we really fine tune the concentration of the antibiotic till you get a perfect flat line over several hours of the number of living cells. Right. It is difficult to do these experiments, you have to find the unit. And when that happens, the total number of living cells is constant. What is your prediction for the total number of dead cells, which is the difference between these 2 curves? Every living cell, every division makes 1 dead cell, and those dead cells keep adding up, right. So, number of dead cells must keep on going up, and not just go up randomly, it must go up linearly, because the rate of making dead cells is simply proportional to the number of living cells, and that is exactly what you see, right. This is in log space, but if you look for the actual number of cells as a function of time, this is the same curve, except this is not log, it is the actual number. Number of living cells is flat, the number of dead cells, which is this minus this, is a perfectly increasing straight line, perfectly increasing straight line, right. So, this is what we have to try and explain. Is there any model of a cell that can capture these kinds of experiments, right. They sort of are making sense, they sort of are telling us there is something going on with division, death and so on, but not completely. Okay. So, now we are going to take the kind of math that we discussed yesterday, right, these random walks in molecule space, and we are going to try and make a model. So, I am going to make literally the simplest possible model you can of cell division. Well, I guess literally the simplest possible model is that the cell is the minimal replication unit. 1 cell makes 2 cells and that is it, 2 cells makes 4 cells, right. But that model cannot capture these kinds of data, as I will show you in a second. Okay. So, this is starting to give us a hint that the minimal unit of replication is not a full cell, it is something less than a cell, right. Something less than a cell can actually self-replicate, and we do not exactly know what that is, but it is certainly not DNA. So, something bigger than just DNA, it will be DNA plus ribosomes plus nucleic acids plus all the enzymes to make those, and all the collection of things need to make it, can self-replicate, and whatever that is less than a full cell. Okay. Since we do not know what it is, I am going to give it a name, we are going to call it a widget. A widget is sort of American slang for an object you do not know what it is. Okay. So, whatever this is not a molecule, it is a collection of molecules, right, but for the purposes of this talk I am assuming it is just like a single molecule, right. It is the minimal self-replicator. Okay. Now, from this point on, this is just a model, its assumptions are, they are of course wrong, right. The question is, does it help us understand what is going on in the data, right. Remember what I said in the first day, you are not making a model to make a statement, this is how you believe the world works. You are making a model to see where your assumptions will take you. So, here is what happens. So, I guess we turn on the light. Let's see if I can do this. No. Well, okay, that will do for now. So, remember yesterday I had this this thing that d n dt is alpha minus gamma n, or m. M is the number of mRNA, right? That was the equation we were using, right? Here we are going to do dw dt, right, is alpha, but not just alpha. So, each widget is a self-replicator. So, the more widgets you have, the greater the rate at which they are made. So, it is actually alpha times w, right, minus gamma times w. So, the difference between these two is here. The rate of synthesis is not, it is not that the widgets like RNA is made from DNA, but widgets are made from themselves. Okay? So, the equations are just slightly different, and that is enough to screw everything up. Right, and you saw the same equation in the population growth models that Malay was talking about on the first day. Yeah. So, this is an easy equation. This is alpha minus gamma times w. And the solution is also very easy. W of t is w of t equals 0, e to the alpha minus gamma t. Yeah. That's here. Okay. So, microscopically though, that is the macroscopic equation, which we know is wrong. We know it is wrong because widgets are discrete, and if they are discrete, they have to be described by the kind of mathematics that I described to you yesterday. And so, we are going to try and make the discrete version of that equation. That is it. Okay. This is literally as simple as it gets in terms of a model of self-replication. So, what happens? With some probability per unit time alpha, one widget can become two widgets. Right? And with some probability per unit time gamma, one widget will get destroyed, because we assume that there is wear and tear and something happens, right. So, when this happens, the number of widgets goes up by 1 and you take a step to the right, and when this happens, the number of widgets goes down by 1 and you take a step to the left. Yeah. Now, that is totally cool, but what happens to cells? Now, empirically, remember we said that we wanted somehow to have a model where the number of cells is the thing we are counting, and cells can divide or they can die, right. So, somehow we want a model where the number of living cells has some division rate, right. Let us not worry about whether it is a microscopic rate or whatever, but some rate, just like this in alpha, there is a rate 5 plus, which is the division rate, and one cell can become 2 cells. And when that happens, the number of living cells is increased by 1, and there must be some other rate where the number of cells dies, but since the dead cells are still floating around in the tube, we can nicely keep track of them. So, the number of dead cells in this process goes up by 1, and the number of living cells goes down by 1. Okay. And if that is what you have, then the equation to describe the total number of cells is very similar. The number of living cells dCl dt is phi plus minus phi minus times Cl, right. That is your standard sort of population ecology model for exponential growth in the absence of a limiting nutrient. And the number of dead cells is proportional to the number of living cells. At least the rate of increase of the number of dead cells is proportional to the number of living cells, because living cells are the ones that die. Right. So, these are the equations that will govern a cell. Now, the important thing to know is this alpha and this gamma we assume are fundamental parameters. They are determined by physics and chemistry. They are constants that depend only on the properties of DNA and ribosomes and self-replicating units. Okay. They might be influenced by adding an antibiotic, right, but whatever it is, in a given external condition, they are constant. These parameters are not constant. They are empirical parameters. They are things you measure. And as we saw, they are not really constant over time, right. All kinds of things happen. Even when you add a poison, the cells somehow manage to survive, then they die, right. So, these phi plus and phi minuses are not constant. These are not constant at all. So, somehow we must have a model or a way to pull out what these macroscopically measurable, well, these biologically measurable properties are from these microscopic properties. And the way you link those two is you make an effective model. You make a model to pull out effective parameters. This is a standard move. It's sometimes called coarse graining. I mean, there are multi-level or whatever it is, right? Many aspects of modeling take the microscopic to the macroscopic. So, there is no reason that there is only one way to do this, there is many, but this one is literally the simplest one I could think of. So, I said suppose a cell just contains a number of these widgets, right, and as a function of time the widgets can divide, the widgets can self-replicate, they do not divide, the widgets can self-replicate or they can get destroyed. And so, what happens is if the widgets self-replicate and you get more widgets, the cell becomes bigger, and we just measure cell size by the total number of widgets it contains, the total biomass. Now, at some point in time, in this case, let us say when the number of widgets hits 5, omega equals 5, then the cell divides, and it divides in such a manner that it just puts a barrier right in the middle, and then randomly if a widget happened to be on the left side of the cell, right, it will go to this daughter, and if it happened to be on the right side of the cell, it will go to that daughter. So, there is a little bit of randomness here also. There is randomness here because the rate of creation is a Poisson process, destruction also. There is randomness here because of division, right. So, this is how division works. If the number of widgets manages to reach a certain threshold, cell instantaneously divides and partitions everything according to the flip of a coin to left or right. What about death? This one is slightly easier to understand. If the cell loses its last self-replicating unit, its very last one, then clearly you can't divide anymore. You can't do anything because you can't make that out of nothing. That is the whole point, right. You can't spontaneously generate life except on billion-year time scales, right. So, cell will die. So, I can take these parameters, I can feed it into a model where they live inside a bag which is the cell, and from these parameters I should be able to guess how often a cell reaches the threshold and manages to divide, or how often it reaches the bad threshold and manages to die, right. And so, my whole move, my whole strategy will be to see, can I guess what these phi plus and phi minus parameters are based on only knowing this alpha and gamma parameter and maybe this thing, the threshold. Question? No, not yet. Okay, so I skipped a line here. What happens is you get 5 widgets, and as soon as you get 5, the cell divides. So I could have put a little line in the middle where there are 5, and as soon as there are 5, it divides. Yeah, good question. So everybody's happy with this? So this is, as a word model, it's very simple, right? Widgets can self-replicate, widgets can die. A cell has a bunch of widgets. When the number of widgets hits 5 or 10 or whatever you want to pick, the cell instantly divides, and when it goes to 0, the cell is counted as dead. Right? Very simple. So the question is how to find out how often this happens and how often this happens. It seems quite complicated. There is so much that could happen, right? But the math that I told you yesterday will help you do it exactly. It's nothing different than what we wrote down yesterday. Okay? So now you are all experts on this, right? So I am not going to belabor the point, but let me unpack it a little bit, right? So what's nice is now you know enough to completely understand where these equations came from. You could write down these equations yourself. You could take the old model, which is dw dt is alpha minus gamma times w, and really work out the consequences. Okay? So here's how you make that model. Just like yesterday, where you had C0, C1, C2, C3 was cells with 0, 1, 2, 3, 4, whatever it is, mRNAs. Right here, C0, C1, C2, C3 are cells with widgets. So instead of calling it Cm, I am calling it C sub w. Instead of calling it C sub k, I am calling it C sub w. Okay? But this does not have to go on forever, because we know that no cell can have more than omega minus 1 widgets. Because as soon as you get omega, as soon as a cell leaves, so let us say there are 600 cells in this bin. As soon as any cell from this bin gets 1 extra widget, instantly it divides. And when a cell divides, what happens? The widgets get partitioned into 2 daughters. But if it has 10 widgets, you do not necessarily get 5 and 5, right? you might get 8 and 2, right. Whatever you get, the sum has to add up to the total, but the distribution will be binomial, because each widget has a 50-50 chance of being in each daughter cell. So, that is why you get omega choose W 2 to the minus omega, that is where this term comes from, right. Now, that is the division. What about depth? If a cell has exactly, let us say there is 50 cells with exactly one widget. If one of those cells happens to lose that widget, then it becomes dead and it gets added to the column of C0, and over time this will go on increasing. So, the total number of living cells is the sum of all these, which is what I have written here. Total number of living cells is the sum from omega W equals 1 to omega minus 1 of CW, and the total number of dead cells is just C0. Okay. Now, what about the underlying rates? This is exactly like we wrote yesterday. The rate at which you jump to the right from the bin CW is CW times alpha times W this time, because yesterday we did not have the times W part. The rate at which you move to the left is exactly like yesterday. It is gamma times W times CW. Right. So, these are very similar as to yesterday, except that this is extra W term. So, these are the rates, the random rates at which cells move. So, a single cell will be jumping back and forth, back and forth, back and forth, back and forth, right. And if a single cell jumps here, then you get 2 cells, and if a single cell jumps here, then you lose a cell. So, if you want to know the effective rate at which cells are dividing and dying, you do not need to know this whole thing. You need to know currently how many cells are sitting here and how often do they jump. And so, this is just C omega minus 1, that is how many cells are here, and the rate of jumping to the right is alpha times W times C. In this case, W is equal to omega 1. So, it is alpha omega minus 1. So, it is alpha times omega minus 1 times C omega minus 1. So, this is the flux of exiting on the right side. The flux of exiting on the left side is very simple, it is just gamma times C1. Right. So, from the microscopic model, we know exactly how many are jumping out from the right and how many are jumping out from the left. But remember our effective parameters, we wanted to write an equation where we could pretend that the rate of cell replication is proportional to the total number of cells, and the rate of cell death is proportional to the total number of cells. So, I just define it, right. So, I say that the flux of cells jumping out to the right, I want to write it just as some effective parameter times the total number of living cells. Even though I know it is only these cells that are at risk of dividing, since I cannot tell just by looking at a cell which of these states it is in, I just know the total number, right. So, I say that the rate of jumping to the right, if I put this blinker on, all I can do is say macroscopically that it is proportional to the total number of living cells. So, that is it. To define phi plus, I say phi plus times CL must be equal to this flux. Similarly, I say phi minus times CL must be equal to this flux. That is it. Question? Oh, yeah, so very good. So, omega need not be a constant, right. So, maybe some cells could divide when there is 15, some cells could divide when there is 8, maybe there is noise in that also, there is some errors, right. So, as I said, this is just the simplest first model I can imagine making. I could, the omega need not be constant, yeah, very good. And there may be no single thing the cell is measuring, maybe if it gets to the right length it will divide, if it gets to the right amount of DNA it will divide, there may be many different, right? So, I never said this model is a complete and accurate model of how cells divide. I am saying that this model is the simplest model I can think of that will couple microscopic things like this to a macroscopic things like that. It contains at the moment the fewest available assumptions, and I can compare it to the experiments, and if this is sufficient, well, that is pretty good. It does not prove the model is correct, but it proves that most of the ingredients you are thinking about, you are on the right track, right? And if it does not work, I have to add the kinds of things you are talking about. Okay. So, this is actually a very important move, and I want you to realize what we have done. We took a model where exactly, because that was the assumption, the rate of creation of widgets is alpha times W, the rate of destruction of widgets is gamma times W. We have now made an effective model where we can pretend that the rate of division of cells is phi plus times CL, and the rate of death of cells is phi minus times CL. The reason we can pretend to do that is because we actually know the exact rates coming out on the right and to the left, because we've solved the microscopic model. And once we've solved that model, we just pretend that I can summarize everything with these effective parameters. That's why they're called effective parameters. They're not real, they arise from some more complicated microscopic world. In particular, suppose this shape changes. Suppose the number of cells on this side becomes lower, the number of cells on this side becomes higher over time, right? Then this phi plus and phi minus can themselves change over time. On the other hand, if this shape reaches a steady state, like we derived yesterday, then phi plus and phi minus from that point will appear constant. They will look constant, but actually they are not. Right? They changed over time, then they themselves reached a steady state. The connection of this to the equations, very simple, just like we had yesterday. dCW dt is these two minus terms and the two plus terms, exactly the same as we wrote yesterday, plus this bit. Okay? This says that every bin has an additional way of getting cells, not from the left or the right, but from here, two cells can just parachute down and land by binomial partitioning. This is the only additional piece, and all this term here is there for normalizing and all that. Okay? Cool. So, what I am saying is, you know this math. Not only you know it, you know how to deal with it, you know how to solve with that. So, in this talk now, I am not going to go and solve with this equation, but it is exactly the same way as we did yesterday. You make a ladder, and from that ladder you assume C0, or in this case C1, and then you figure out the shape of this. Now, yesterday the shape was a Poisson distribution, but this is a slightly more complicated model, so there is no reason for it to have a Poisson distribution, but it will have some shape. Okay? Now, just to cut to the chase, let me show you the comparison of the experiments with the model. Okay? These are the experiments. I can plot either the total number of cells or the number of living cells, just like I showed you earlier. Right? And I can do this for various levels of the antibiotic. And for the different levels of the antibiotic, I know the total number of cells. So, this is where the food runs out. So, do not look at anything in this gray region. When there is no antibiotic, you get exponential growth. When there is more antibiotic, the exponential growth slows down. At some point, the total number of cells becomes constant because cells are dying. Right? With no antibiotic, you get exponential growth. At a perfect concentration, which is called the minimal inhibitory concentration, it becomes flat, and then with more antibiotic, we get perfect exponential decay. But you get this transient. Okay. So, it is not a constant rate of division and decay. Right? Now, here is the data. Now, what is the fit? What have I fitted? So, in principle, this model has how many parameters? 3 parameters, right? But by just looking at units of time in one of the experiments, you can set one of the parameters. Right. So, it really has 2 effective parameters. One effective parameter is the rate of decay versus the rate of replication of a single widget. And one assumes that that somehow is related to the antibiotic level. So, that I can pretend is reflective of the antibiotic level. The other parameter is this omega, which I have assumed is constant. Right. And in this particular case, I have just picked omega equals 10 as a rough estimate. Right. So, this is not a fit. I have just kept omega constant, and then there is only one parameter left, and that parameter, if I sweep that parameter, I get qualitatively very similar behavior for the total number of cells. And more interestingly, for the number of living cells, I exactly replicate this transient. Right. So, even when I add the Poisson, initially the rate of division is still pretty good, then it goes down. I will explain to you in a second why that is working. Right. So, I completely replicate all the qualitative features of the data. On a log linear plot in 0 antibiotic, I get linear growth. At very high antibiotic and after the transient, I get negative linear growth. And at this particular value MIC, where gamma equals alpha, I get a flat line. Right. And this is where you can now compare with an even more simple model of a cell. Now, suppose I assume that the cell itself was a minimal replicating unit. The whole cell is needed to do minimal replication. Then what would the value of omega be? The whole cell is the minimal replication unit. So, what is the value of omega? It's 2, because as soon as you get 2 copies, the cell divides. So, any given cell always has just one widget. Okay. So, I can ask, if I assume that the whole cell is the minimal replicating unit, what would the model predict? And here is what it predicts. It predicts there is no bump. To get a bump, you have to assume that cells have more than one replicating unit. So, this is sort of real world suggestive experiment that says that the replicating unit is smaller than a single cell, bigger than DNA, but smaller than a single cell, right? Because if you assume it is the whole cell, this is what you get. So, of course, I am covering up a lot of subtleties here, but that is roughly the idea. Now, let me explain why you get this bump, right, and why you get different rates of division and death and so on. Okay. So, this is interesting. So, this now contains everything that the model contains. Okay. So, this is a full comprehensive result of the model, and here let me go to the board again. Okay. So, what have I plotted here? These are those histograms. Now, in the histograms, for the sake of clarity, I have not plotted C0, which is the number of dead cells, because after a while C0 will become very, very big compared to the total number of living cells. Okay. So, I have only plotted the living part, which are cells that contain one or more widgets. Over here, I have plotted the histograms for a model where I have assumed big omega is 10. And over here, I have plotted the data for a model where I have assumed that big omega is 50. Okay, just to get you thinking about it. You know that the cells that divide are the ones that are jumping out from the right side. So, all else being equal, the more the height of the rightmost bin, the faster the rate of division. The smaller the leftmost bin, the slower the rate of death. Okay. So, in this condition, which is where gamma, which is, remember, the decay rate of the widget is 0, it is like 0 antibiotic. All the cells are dividing, none of the cells are dying. Okay. So, everything is a phi plus, nothing is a phi minus. Okay. Now, as I add antibiotic, the height of this bin, the cells that are just ready to divide, go down. It is not that these cells are static. Okay. You have to remember, the cells are constantly jumping up and down here. They are constantly going up and down. What you see here is just the steady state, but underneath there is a constant flux, right. They are going back and forth, back and forth, back and forth, and randomly one will jump. Okay. So, this phi plus goes down, and then you suddenly get a phi minus, right. There are some cells in the leftmost bin. It is less than 1/3 of the total number of cells, but they are there. And after a while, phi minus becomes bigger and bigger, because most of the cells are almost at the boundary of death, and very few cells divide. Okay, that is in this case. So, here you have the effective division rate is coming down, the effective death rate is going up, but under these conditions you can find a situation where in the same population some cells are able to divide and some cells are able to die. Okay. But by the time you dial this omega up to big number 50, then you find that the division rate goes down and it goes to 0 before any death starts. Okay. And then the death rate goes up separately. Okay. So, if I were to take all this data and plot it, it comes into this plot, and let me just compare this plot first to what the, if you pull out any microbiology textbook, this is what you will see, right. This is the antibiotic concentration, right, and this is the rate of division, and this is the rate of death, right. And the way those textbooks will explain these curves is they will say, I plotted the log of the number of cells as a function of time, and whenever it is positive, I assume that this is phi plus, and whenever it is negative, I assume that this is phi minus. right. So, they assume that whenever you see growth, there is only division going on, and whenever you see decay, there is only death going on. I mean, this is what the textbooks show. So, if this is what you see, then what would you, as a function of antibiotic concentration, what would you expect? Well, right, you would expect that as a function of antibiotic, the division rate will just slow down, and then at some point it will stop dividing, and correspondingly the death rate will be 0, and at some point it will start dying further and faster and faster, right. And at this point, which is a special antibiotic concentration, which I have called minimal inhibitory concentration, right. According to the textbooks, this is the bacteriostatic condition. It is called the bacteriostatic condition. So, according to the textbooks, you should get no division and no death. That is why the total number of living cells is constant, because they are all there and they are still alive, they are just not dividing or dying, they are static. Okay. So, when you look at this model, what does it actually show? I can plot the division rate, which I have plotted on this side. The division rates are all the curves that are going down as a function of antibiotic. I can plot the death rates, which are all the curves on this side. The death rate curves are the ones that are going up as a function of antibiotics. Okay. So, all these are the division curves, all these are the death curves. Right. All those curves will intersect, and wherever they intersect is when you have the balance condition. Right. So, in this case they intersect here or here or here or here or here. The different colors represent doing the test at different values of this big omega. This is when a cell divides with 10, this is when a cell divides with 50, you could do it with 2, 3, 5, 10, 50. Okay. 2 means that the cell is the minimal replicating unit. And what you find is very interesting. What the textbooks call the bacteriostatic limit, or the cell stasis limit, in this model is not static. Right? You can actually get a non-zero rate of division, you can get a non-zero rate of death. It still looks like the cells are not growing because the number of living cells is constant, but we know the number of living cells is not constant, right, from the data. This is the crucial experiment. According to the textbooks, if I had done this experiment, if it worked the way they said, the total number of living cells would have been constant because they are not dividing or dying. So, the total number of dead cells, there should be no dead cells at all, right. So, the total cell curve should also be flat, and it is not flat, and I can actually measure this. I can measure the rate. If I can measure the rate and compare it to this, I can actually figure out what the effective value of omega is. Okay. So, in a world where there is randomness, so this is a very non-random world. This is the world where the number of widgets goes to infinity, and then all your fluctuations come down, just like we have heard. When you have large numbers of things, the fluctuations are low. So, if I actually plot the height of the intersection point, which is the independent rate of division and death when they are both balanced, as a function of this omega, you find that it goes to 0 smoothly if omega goes to infinity. This is 1 over omega, right. So, this is what is a very noisy limit, high fluctuation limit. This is the low fluctuation limit, which is what is in the textbooks. So, textbooks are not obviously wrong, but they are only considering the low noise limit of what is a true model. In particular, for our experiments, it is not that limit. And what we find is omega is anywhere between sort of 5 and 10. Okay. So, by making these measurements of the random rate of division and death, you can estimate how many self-replicating units there are in a cell, and we are saying it is between 5 and 10. Okay. Now, some much more complicated experiments have been done by the group of Jun et al., where they measured nutrient amount, ribosome amount, they measured replication forks, they found an effective scaling law, they found an effective collapse, all kinds of things, right. And in a totally independent way, they figured out that under different growth conditions, there is anywhere between 2 and 8 minimal self-replicating units per cell, right, which is roughly the same order of magnitude that we found by independent means. Yeah. So, this really leads me to believe that we are on the right track, right, that there is some notion of self-replication which is smaller than a single cell, at least abstractly, and that a cell contains more than one of these. And how do I know it's more than one? Because I have to get rid of every last one before the cell dies. That's how we managed to do it. We managed to find it because you can destroy half the cell or 1/3 of the cell, or sorry, 2/3 of the cell or 3/4 of the cell, and the cell still managed to divide. So, clearly they still contain self-replicating units. Here they have done it a totally different way and they get the same number. Okay. So, it is nice. I think I will stop 10 minutes early today, good. So, you can read about this, it is published, but I just want to say that the way this happened is to me it is one of the most fun projects I have worked on in a long time, because it started off with, as I was trying to tell somebody over lunch yesterday, if you are working on something, you have to be working on something that is a puzzle, it is mysterious to you. You should not be saying, I am going to cure cancer, I am going to make a model and get all the parameters and put them in, right, because you do not have any expectation going in. You should be working on something which, based on everything you know, it does not make sense. If you are not working on something like that, you have no compass. How do you know you are making any progress? So, once you find a puzzle, to me the thing that did not make any sense was this exponential decay of bacterial cells, right, and I made a little model, and the little model literally took one evening, right, that entire, because that equation takes no time to write down, right. So, that entire thing took one evening to write it down, solve it, get the predictions, everything, okay, took literally one evening. And then to do the careful experiments, right, but now that we know what we are looking for in the model, we wanted to separate living cells from dead cells, and I had this fantastic master student from BITS, Soumya, and she really calibrated the division and depth measurements to the point where we get these curves with very low error bars. I do not know if you noticed, but all those curves had error bars, right. So, it was replicated, but it was small enough, right. So, this is Ashwin, who is the microbiology expert on the NCBS campus. So, he was the one who chose that particular antibiotic and set up the correct growth conditions, right, and he was the one who helped us actually flesh out the experiment in a way that was doable. Yeah. Suman is a theoretical physicist. So, it is with Suman that I worked out all the aspects of what that model was telling us, right, and Savita is a postdoc in Ashwin's lab, who is the one who mentored Soumya and really taught her how to get the experimental design correct, right. So, it is a proper experiment theory collaboration. It is not that the experimentalist had something and then came to the modeling people and said, look, just give me an equation that fits all the data, right. That is wrong, right. There was a puzzle which we both agreed was puzzling. There was a little model which was not a complicated model, but the bulk of what makes this a valuable project is that the experiment was done well. It was not me, it was all these people, right, and collaborators from all over the place. So, I will wrap up. So, I typically show this. So, Freeman Dyson, remember I mentioned him, he is self-replicators. So, this is not my last, I am going to lecture later, but this is it is a nice way to close everything that I have been trying to say. Somebody came up to me at lunch day before yesterday and said, I do not understand what you are trying to get at in these lectures. I mean, are you trying to tell us different ways of modeling or whatever? I am not trying to get at anything. I am just trying to give you an overview of the different ways you can work, right, and to show you that the math is not that complicated, but also you should not really take the math too seriously, but it is a fun place to be. If you happen to be skilled in mathematics, right, then you have access to a different way of thinking than a traditional biologist. I am not saying it is better or worse, but it is different, and therefore, you might have insights that they do not, and vice versa. Right. So, I will just read. So, Freeman Dyson was one of the people who put together this theory called quantum electrodynamics or quantum field theory, which is the most successful theory in all of science. So, he knows what theories are all about, right. So, this is a recent interview. So, he says in those days, so he is talking about the 1960s, I guess, people became physicists in those days. Those kinds of people tend to become biologists, in the sense they become interested in biology. A lot of people who are really computer scientists are doing biology. The line between mathematics and biology is not so sharp. I would say that is where the young people are. They are having the same good times now that we had 50 years ago. So, you are in a good place. Yeah, it's a good time to be a person bringing mathematics to biology. But what that means is you got to bring it with the level it deserves. You shouldn't be coming in as sort of handmaids to some experiment and just try and chalk out some 6,000 parameter differential equation. That's not your job. If that's what you're doing, don't do it, right? And please don't try and cure cancer. Nobody in this room is going to cure cancer. It's just not going to happen. You don't have the skills. To cure cancer, you're going to have to go out into a hospital, work with patients, pull out cells. That's not what we're doing, right? Paul Nurse is a biologist. Freeman Dyson is a physicist, right? Paul Nurse is an actual biologist. He's a Nobel Prize winning biologist, right? So he says the idea is to look for ways that can transform all this detailed mess, molecular interactions, biochemical activities, biophysical mechanisms, into logical informational structures and processes, right? So he's talking about abstract. This will lead to the understanding of a cell as a computational machine. This approach will shift biology away from the common sense and familiar world, right? So many biologists use common sense intuition to guess what's going on, and a lot of the time they're correct, but sometimes it leads them astray, right? Lead them away from the common sense and familiar world that has generally occupied the past into one that is more abstract, which is where you guys have a serious comparative advantage. So if you're not really pushing that aspect of your skills, you're not contributing, okay? And you should not get stuck in a rut where you're doing a routine modeling, simulation, dare I say, it's machine learning, which is, totally ridiculous, right? That's not going to get you anywhere. You have to have insight, and you have to put the insight into a model, and they can be extremely simple models. The models we've seen in the last week are all extremely simple, but they give insight. And if you're not providing insight, you're not actually doing anything, okay? So I'll leave with a little plug for the place where I work, right? So NCBS, we have a little group of people called the Simon Center, where we have so Madan, Shashi, Sandeep, me, Shachi. We work on molecules, cells and cell populations, networks in my case. Madan works on biophysics of a cell, and Shashi builds experiments at the entire population level and for non-biological things. And here is the actual page of, I was sitting in Delhi University and I couldn't sleep, so literally the actual page where I realized how you could get exponential decay. So I started on top, and by the end of it I had the full equation. It's all done. This is the entire thing, right? I didn't spend years making this model. Okay, so it's that easy, so you can do it. Okay, so we'll stop now, 5 minutes early, because I took 5 minutes extra yesterday. And now you guys can, now what would you like for the evening lecture? We have an evening session, right? I know you're all tired and saturated, so I don't want to give more data. We can just have an open-ended discussion. I can do a tutorial on how to do a certain calculation again. I could give another talk on a completely different subject. So what would you like? Any votes? Right? We're not going to cancel the session if you think that's what's going on. So go to the beach or something. Yeah. So how many people vote for a tutorial where I do a calculation in more detail? Nobody wants that. Okay. How many people want just a philosophical discussion? Oh my God. Okay. Yeah. Okay. And how many people want to see a talk on a different case study, like a talk containing data and so on? A couple of people. Okay. So 3, 4. Well, maybe we can mix it up. Let me take a call. Okay. Fine. So let's take a break and come back for Kavita's talk.